Dear students, today we will be discussing about Mendel's law of heredity. The study of genetics is incomplete without a proper understanding about the laws that has been put forward by the Mendel. So now let's have a brief introduction about Mendel. Mendel was an Austrian monk who was basically a student of science and mathematics. He predicted that how traits are transmitted from one generation to another generation, and Mendel studied these traits in pea plant. So after his experiments of monohybrid and dihybrid crosses, Mendel concluded that there are certain factors that are passed from one generation to another generation. But Mendel explained this phenomena with the help of his first law in genetics. The first law in genetics is called as law of segregation. And to understand this law, first we need to understand the concepts of few genetic terms such as alleles, homozygous, heterozygous, phenotype, and genotype. So first have a look on what is allele. And to understand about the allele, let's revise the concept of a gene. We know that the genes are basically a unit of heredity that carry genetic information. So alleles are nothing, but they are just the different forms of genes. So as you know that alleles are different forms of genes, let's have a look on the simple example to understand allele. For example, we have these two chromosomes, and here we have the genes of the character height, just like for an example. So now there is a question, which genes could possibly present here? We can have either both capital T, or we can have both a lowercase t. So we can also have one capital T and one lowercase t as a gene set. So here the individual with capital T, that is a single gene, is nothing but the allele. So the alternative form, that is either dominant or recessive, these are called as allele. Or you can say in simple words, or in other words, one single capital T or one single lowercase t is an allele. So now what is homozygous? Homozygous can be explained as an individual with two identical alleles. So now what is heterozygous? Heterozygous can be explained as an individual with two different alleles. So now let's talk about the phenotype and the genotype. The phenotype can be explained as the observable parameters of some organism. For example, the color of skin, size, forms, and structure. These are known as a phenotype. The phenotype can be in shape of tall-headed plants, short-headed plants, or if you talk about the seeds can be round, the seeds can be wrinkled. And if you talk about the genotype, genotype can be explained as the genetic composition of an organism, but the basic difference between genotype and phenotype is that the genotype cannot be seen on visual basis with the help of naked eye. So now that we know what alleles are, let us try to understand the laws of segregation. The law of segregation can be explained that during gamete formation, the allele for each gene segregate from each other such that the each gamete carries only one allele for each gene. So now we will have an explanation of this law. So to understand this, let us take a simple example. For example, we have a single cell having two chromosomes in it. These chromosomes have a set of genes, that is a capital T and a lowercase t. The capital T represents for tall hybrid plants, and the small t, or you can say lowercase t, represents a short hybrid plant in an individual. And remember that the first part of the law of segregation says during gamete formation. So this is the process of meiosis, and as you can remember that meiosis is the type of cell division which gives us four cells with half number of chromosomes. So the second part of the law of segregation says the alleles of each gene. So here we are referring towards a capital T and so the next part of the law of segregation states that segregate from each other. This means the alleles will separate. They will not remain together in the process. And now comes the last part, which says that one gamete carries only one allele for each gene. Now, this part means that at the end of meiosis process, we will get four cells such that each cell will have only one allele in it. So here, two cells will have capital T and the other two cells will have a lowercase t. Thus, each gamete gets only one allele. So all this phenomena was hypothesized by Mendel and later it was accepted as a law of segregation. So now let's talk about law of independent assortment. The law says that genes for different traits assort independently during gamete formation. So now let's have an explanation of this law. Let us understand this with the help of this cell. The first part says that genes of different traits. This means that we are addressing the genes which represent different parameters. So here we have gene sets capital A, capital B, small b, capital C, small c on these respective chromosomes. So these are the genes of different parameters. The second part states a sort independent of each other. This means that the genes will not have a fixed pattern to follow when they segregate in the cells. So here the alleles of the gene set capital A small a will assort independently to the other two gene sets. And remember, it is not mandatory that all the dominant alleles will get assorted in the cell together. So the allele capital A can get assorted with any allele from the gene set capital B small b or you can say capital C small c. So that is the reason that why we get the gametes with different possible combination at the end. The assortment of allele is purely random, remember, and which does not have a predictable or you can say fixed pattern during the process of gamete formation. So now there is a question that which type of cross will help us to understand the law of independent assortment. So the answer of this question is dihybrid cross. So if it is a monohybrid cross, then there will be no independent assortment of different alleles because in monohybrid crosses we are considered only one parameter at a time. So we will be dealing with only single gene set. It can be either capital T that is for tall headedness or it can be a small t which is for the short headed plant. And thus law of independent assortment is better understood when there are more than one parameter to be studied at a time. So dihybrid crosses can be explained that the mating that involves parents that differ in two traits. As you can see on your screen, there are two parents. In one parent, the flower color purple has a dominant nature and the flower color white has a recessive nature. And there is another parent which is having a tall headedness and the tall headedness was governed by a capital T and the second phenotype is a small headedness which is governed by the recessive small t. When these two have a cross with each other, the F1 generation will always be a capital P, small p, capital T, and small t, which can be explained that the F1 generation will be having you can say a tall-headed plant having purple color flower, and this is also known as law of dominance, which will be explained in our future studies. So if we make a cross of the F2 generation in a dihybrid cross, then we will be getting a 9331 ratio, which means among the crosses which have been made, the nine, offspring will be tall heighted having purple color flowers. The three will be tall heighted having white color flowers. The other three might be short heighted having purple color flowers, and the one would be a short heighted and white color flower. So, if we talk about the last law of genetics, which is the law of dominance, this law states that some alleles are dominant while others are recessive. An organism with at least one dominant allele displays the effect irrespective of the presence of the recessive one. As you can see on your screen, that a purple color is having a dominant nature, which is governed by the dominant alleles, and the white color flower, which is governed by the recessive genes. When we have a cross of this kind of plant, then in F1 generation, every plant will be having a purple color flower, because remember, purple color was dominant. So if we see the definition, that an organism with at least one dominant allele will display the effect. If there is only one dominant allele, then the color will be purple. Or you can say if the both alleles are dominant, then definitely the color will be purple. But if there is only one dominant allele, then the color will also be purple in F1 generation. But if we move in the next generation, which is F2 generation, which is also called as F1 called as first filial generation, and F2 called as second filial generation. So in second filial generation, we can have we can have you know a white color flower because of the recombination. So this is the overall concepts of the laws of genetics, which has been proposed by the Mendel. And the Mendel's laws are, you can say, that law of independent assortment and law of segregation. And the third law of genetics is the law of dominance.